Welcome everybody to our February meeting of the New York City Neighborhood Naturalist Group. My name is Lydia Paradiso and I'm a doctoral student at the New York Botanical Garden and a graduate fellow working with the New York City EcoFlora project. The New York Botanical Garden created the New York City EcoFlora project back in 2017 with a grant from the Institute of Museum and Library Services. Building on the garden's historical strengths and collections with emerging opportunities in digital technology, the EcoFlora is a community science project to document the wild flora of New York City. To date, nearly 24,000 observers have made almost 700,000 observations of plants and their biotic relationships in the city. You can explore these observations on the project page in iNaturalist, which I will link in the chat in a minute. Each month, we also sponsor an EcoQuest challenge, which encourages New Yorkers to observe a particular species or a genus and their ecological partners and post them on iNaturalist. The EcoQuest for February is entitled Search for Birch. Birches are small to medium-sized trees that are found on temperate zones in the Northern Hemisphere and are used extensively for their wood and their bark. New York City is home to 5 native species of birch, which can be recognized in the winter by their bark, buds, and characteristic scent. 2 of the common species are gray birch, Betula populifolia, and sweet birch, Betula lenta. There's also 3 less commonly seen species, river birch, Betula nigra, yellow birch, Betula alleghaniensis, and paper birch, Betula papyrifera. Of course, when I say less often seen, this is in the wild, because a lot of these are more common planted species. Our talk tonight will discuss some of these co-occurring species and the factors which have influenced their current distribution and their population genetic structure. It is my pleasure to introduce our speaker on this topic, Dr. Ashley Thomson. Ashley obtained her PhD in 2013 from Concordia University and then went on to complete postdoctoral studies in genomics of Lacethedaceae at the University of Michigan. Since 2017, she's been an assistant professor of forest genetics at Lakehead University in Canada. Her research focuses on tree genetics and climate change. In her spare time, Ashley enjoys cross-country skiing and gardening with her son, Manny, and her husband, Sebastian. She also enjoys spending time with their 2 spoiled fur babies, Nibbler and Bella. Please feel free to enter any questions or comments into the chat during the talk, and at the end we'll go through as many as we are able. You can also at that point you can use the raise hand feature if you want to verbally ask your question. Also, if you're unable to stay for the whole talk or you would like to share with others or view it again, we will have a recording posted on our YouTube page, and also there we have recordings of our other recent talks. So now, without further ado, I'll turn it over to Ashley.
Okay, thanks, Lydia, and nice to sort of meet all of you virtually. I wish I could be giving this talk in person and meet everybody face to face, but happy to be able to give it via Zoom nonetheless. So the talk that I'm going to be giving you today is based on my PhD research, which, as Lydia mentioned, I completed at Concordia University, so that's in Montreal, Quebec. And yes, I think I'll just leave it at that, and we'll move into giving you some background on the study. Oh, I should add, if any of you do want to ask questions throughout, you can put up your hand or enter it in the chat, but I also don't mind. I'm sometimes bad at noticing the chat function as I'm talking, so you're also welcome to unmute yourself and just interrupt me. I'm used to dealing with undergrads who interrupt me all the time, so it doesn't bother me, so you feel free to interrupt. So the talk I'm going to be giving today has 2 main parts. So first of all, I will give you an overview of a phylogeographic study that we did of Eastern North American birches that used chloroplast DNA markers. And then secondly, a second study that followed kind of from that first one, where we examined then introgression, which is basically gene sharing between species at nuclear DNA markers. So starting with the phylogeographic study of the Eastern North American birches. it always warrants giving everybody a little bit of a background as to what is phylogeography. So I've given this talk to colleagues, to students, to various groups, and I mean, really, other than close colleagues, for the most part, many people don't know what phylogeography is and what the point of this discipline of biology is. So when we talk about phylogeographic studies, phylogeography is just a subdiscipline of biogeography, which is concerned with better understanding patterns of genetic structure in usually or often temperate species. And one of the primary questions that phylogeographic studies often ask is how genetic patterns are influenced by historic and demographic factors. So historic factors like Pleistocene glaciations, and demographic factors like past population bottlenecks. So in temperate trees, like the North American birches, phylogeography generally focuses on the influence of historic glaciations, and also more recently, the influence of intergression between species in shaping observed patterns of genetic variation. And the reason that the focus is on glacial history as a evolutionary kind of force or driver in temperate tree species is due to the fact that continental glaciations had very significant effects on the ranges of temperate tree species, and therefore also affected their population genetic structure. So continental glaciations during the Pleistocene epoch resulted in repeated expansions and contractions of species ranges. And so most temperate tree species persisted in just a few isolated pockets of suitable habitat, known as refugia, at the time of the last glacial maximum, which was about 20,000 years ago. I was going to say 2,000, that would be very wrong. So between 18 and 20,000 years ago. And because populations or species in general had retreated southwards and were occupying much smaller geographic areas than their present distribution, that has some really important genetic effects. So reduced population size and significant, contractions of population size, which are referred to as population bottlenecks, can result in significant loss of genetic diversity through loss of rare alleles and associated loss of heterozygosity. And also persistence in isolated pockets can lead to increased differentiation among subpopulations due to the action of random genetic drift. So populations occupying separate glacial refugia are expected to become significantly genetically differentiated simply due to random chance. So, and what studies have shown from both Europe and North America is the number and location of refugia that species occupied is a significant predictor, or significantly associated with contemporary levels of genetic diversity, and also genetic structure, or the degree of differentiation among populations. And there's a number of studies which show that. The one by Aguinaldo from 2005 was a really nice one from Europe, looking at the influence of different life history traits of woody plants on genetic diversity. Sort of a modern take on the traditional study by Hamrick and colleagues, where they used allozyme markers to identify the relationship between various life history traits and population genetic structure and diversity. Oh, too far. So after the last glacial maximum, post-glacial recolonization occurred, and the pattern and timing of post-glacial recolonization in different species influences their contemporary levels of genetic diversity. So after the retreat of glaciers, there were obviously large areas of open habitat that were available. And it's generally believed that populations expanded rapidly from glacial refugia, initially very rapidly, and subsequently population expansion occurred relatively slowly. It's generally believed that some of the initial colonization, recolonization occurred via long distance seed dispersal in woody plants. And this long distance seed dispersal should result in founder effects on genetic diversity. So founder effects are basically when you have, say, like a more large continuous population, such as the refugial population, which is relatively large. And then you have just a few individuals or a few seeds which migrate over long distances, say, dozens of kilometers, so far that much farther than their normal rate distance of seed dispersal. And because just a small fraction of the original population disperses and occupies that new habitat, only a fraction of the original genetic diversity would be represented in that new population. So that's a founder effect. So studies from Europe have generally shown declining genetic diversity moving northwards away from refugia, and that's associated with successive founder events during post-glacial recolonization. However, there are quite a number of, I was going to say recent studies, but again, at the time I should preface that by saying that at the time I started my PhD, the paradigm was sort of that most species should have been confined to isolated southern refugia. There were started to be around 2010, 2011, a few studies from Europe and North America suggesting that a number of boreal temperate tree species, or a few examples of boreal temperate tree species, actually occurred more widely distributed during the last glacial maximum than would have been indicated by fossil pollen records. So for example, beech, Fagus grandifolia, a molecular genetic study by McLaughlin et al. I should have included the reference here. Can't remember the year of it, but it was fairly groundbreaking at the time, in that they showed relatively clearly based on molecular markers that populations should have persisted at relatively low density, but across a fairly broad range south of the ice sheet margins and close to the ice sheet margin during the last glacial maximum. So that had some very important implications for our understanding of how fast trees might actually migrate in response to climate change, because previously, if you look at the paradigm where all species were confined to isolated refugia, then the outcome would be that the calculation of how fast species were able to migrate is wrong if you look at that they migrated a shorter distance from just south of the ice sheet margin versus a longer distance, say, from the southern Appalachian and other southern refugial areas. So what that study showed is that that tree migration rates might under climate change might actually be much lower than what we previously expected, because species may in many cases have been located much closer to the ice sheet margin than what we had previously thought. But there are quite a number of studies now that show that overall phylogeographic patterns don't fit just one model, that phylogeographic patterns vary significantly among species, especially in North America, and that those patterns are strongly influenced by cold tolerance and dispersal capacity. So generally, cold tolerant species have higher genetic diversity because they were able to persist across a wider range during the last glacial maximum, and so would have had larger effective population sizes and lost less genetic diversity due to genetic drift. And then also dispersal capacity. So dispersal capacity has been strongly related to levels of genetic diversity in a number of studies, and that's thought to relate to contemporary gene flow, but also how fast species were able to recolonize after glaciation. So dispersal ability has been shown to strongly affect patterns of genetic diversity at chloroplast markers. So, and I've just shown some examples here. So obviously, you guys will all know that this is an acorn from an oak species. Don't ask me what oak species it's from. I just grabbed it off Wikipedia. Actually should have written that down, especially for this group. And then here we have a birch, Samara. So obviously these two different groups have very different seed weight and very different dispersal capacities. And as such, we expect relatively stronger differentiation among populations with lower dispersal capacities like oaks than compared to species that have longer distance dispersal capacity like birches. And that's because these populations with larger seeds should have less gene flow and should have a shorter colonization distance, which results in more significant founder effects during post-glacial recolonization. So the study that I did for the phylogeography of the Eastern North American birches is what's called a comparative phylogeographic study. So normal phylogeography studies look at patterns of genetic structure in just a single species. But more recently, studies that compare phylogeographic patterns among several co-distributed species have become popular for a number of reasons. And one of those is that studies have shown that there can be a very significant influence of introgression, or basically allele sharing, allele capture between species, resulting from hybridization. So by looking at phylogeographic patterns among multiple co-distributed species, you can determine the influence of introgression on the phylogeographic patterns that you're observing, and that helps to reduce potentially erroneous inferences about phylogeographic history of a species. Also, comparative phylogeography, if you look at co-distributed, especially congeneric species, can help to control for shared life history traits. So it controls for differences among dispersal capacity, so that any differences in phylogeographic structure that you observed should be better explained by differences in post-glacial history and recolonization than by differences in life history traits like different mating systems, etc. So birches are the group that I chose to do my PhD work on. It's a bit of a long story why I landed on them. Originally, I actually wanted to study yews in the Himalayas, but I got a little bit freaked out when I heard that I was going to have to be working in the remote parts of the Himalayas by myself. And then I heard about my lab mates who are from that region were telling me about land leeches. And in hindsight, I don't know if they were making it up just to mess with me. But in the end, I decided I would probably stick to North America, where I was a little bit more comfortable with the ecology and doing my field sampling. So birches, of all of the North American tree species that I could have worked with, I selected them based on the fact that they are widely distributed and economically and ecologically important species in North America. But they had a number of sort of good features that made them nice to work with on a comparative phylogeographic basis. So the Eastern North American birches have generally similar life history traits. So they're all woody perennials. They're monoecious, which means, you guys will probably know this, male and female flowers occur on the same plants. And they have wind dispersed seeds and pollen. So that controls for variation and dispersal ability among species to a large extent, as opposed to if you were comparing oaks versus birches. Another thing about birches, and I mean, I knew this, but I guess I didn't really realize how much it would impact my study really before I got into it, was the fact that birches have really complex taxonomy and cytology. And that's because they hybridize quite frequently, and also because of that hybridization, they have frequently associated polyploidy, which results in many cases from hybridization. So they were kind of an ideal model species to work with, and they're just a group of species that I really enjoy. I live in northwestern Ontario, and paper birch just occurs everywhere here, and I grew up always looking at paper birch, and they're beautiful. I think we can appreciate them just from for their ornamental values. So they had a spot kind of near and dear to my heart. So then before I get into kind of what we found at the molecular level, one of the things that I wanted to touch on was what we already knew about the fossil history of, or sorry, the Pleistocene history of birches in Eastern North America. So many of you may be aware that fossil pollen records for many Eastern North American taxa, well, for many plant taxa in general, cannot distinguish to the species level. They can usually distinguish to the level of genus, but they cannot determine, for example, that this is where yellow birch occurred during the last glacial maximum versus this is where paper birch occurred. Now, there are some exceptions to that, but for birches, fossil pollen cannot reliably distinguish to the species level. So our understanding of the glacial history of birches is really the history of birches in general, not individual species. So this is a map of the fossil, or sorry, basically the occurrence of fossil pollen of birches between about 18,000 years ago and more or less present day, or present day at the time that this study was put together. Oh, sorry. This is a study by, I think, Steve Jackson et al. I believe that he's a researcher in Wyoming. His group has done a lot of work on really fossil pollen group of a number of different species. So at the time of the last glacial maximum, the fossil pollen data indicates that there was one refugium, one full glacial refugia for birches that occurred sort of in the central to, yeah, around the central Appalachian region. So this line in this diagram indicates the southern extent of the ice sheet margin. And then the shading basically indicates the frequency of birch pollen. So the darker the shading, the more frequent the birch pollen was in those, I believe they did sediment cores for this study. And then around 15,000 years ago, you can see a few other, one little other spot here, kind of around the Western Great Lakes region, where there seems to be an additional, possibly low density refugia for birches. And this just depicts then how populations expanded. So before we do a phylogeographic study, which relies generally on molecular markers, we always, or I shouldn't say always, but probably 99% of the time, people look to familiarize themselves with what is already known. And that's because the molecular patterns that we observe cannot be really interpreted just on their own. We rely on some of this fossil pollen information to really make sense of the patterns that we observe and interpret it in a meaningful way. So yes, I wanted to include that because it will help to illustrate some of the patterns that I'm going to show you. So the three species that I chose to look at for my work were paper birch, yellow birch, and sweet birch. So you guys have looked at this now in your search for birch, which is great, but just a little bit of a background and a few things that you may not be as familiar with. So paper birch is a hexaploid species. So it doesn't have a regular diploid chromosome number of 28 as a normal diploid birch would have. So instead of having 2 alleles at each locus, it has 6 alleles. So there's a lot of different potential genetic variation that can occur in the species because one individual can have up to 6 different alleles at each locus, as opposed to a normal diploid individual, which you guys would probably be more familiar with from Mendelian genetics, can have just 2 copies of any, or sorry, 2 different alleles at any particular locus. So paper birch occupies a really wide variety of habitats. It's a transcontinentally distributed species, occurs from Newfoundland and Labrador west to British Columbia, the Yukon, and Alaska. And it is the most common birch species in my neck of the woods. So just so you guys are aware, I'm in Thunder Bay, which is also the kind of informal name for our city is Lakehead, and that's because we're kind of at the head of Lake Superior here. So it's a more boreally distributed tree species than the other two, yellow birch and sweet birch, which I'm going to show you. Compared to yellow birch and sweet birch, paper birch has a relatively low shade tolerance. It's generally classified as an intolerant tree species, and it is relatively fast growing and short-lived compared to the other two species. So Betula alleghaniensis, or yellow birch, so like paper birch, yellow birch is also a hexaploid, so again can have up to 6 different alleles at each locus. It's a mesophytic species, so it likes generally moisture, richer sites than does paper birch. And it's classified depending on the source you look at, generally as intermediate in shade tolerance. So it can persist in the understory or relatively shady conditions for a number of years, but it's not completely shade tolerant in that it can survive in the understory for an extended period of time. It's relatively slow growing and long-lived, and it has a distribution really throughout the southern Canada and the northeastern United States, and reaches its highest biomass in the Appalachian region. And then sweet birch, Betula lenta, is the only diploid of the 3 species that I looked at. So it has a diploid chromosome number of 24. Compared to the other 2 species, it's the most restricted in distribution. It occurs really throughout the Appalachian region in the northeastern US. And it occupies relatively similar site conditions to yellow birch, like generally moist, cool sites. I usually found yellow birch and sweet birch growing in relatively close proximity to one another. So just an overview of the methods. So for this study, I used DNA markers, and in particular, I used, I'll get to that in a sec. Well, I may as well talk about it now. Sorry, chloroplast DNA markers. So some of you may be aware that in plants, there are 3 different genomes. You have your nuclear genome, which is what you normally think of as your chromosomes inside the nucleus, thus nuclear genome. You also have the chloroplasts have their own genome, which is a small circular DNA molecule, and then also mitochondria have their own genome. So chloroplast markers have, at least historically, been the marker of choice for phylogeographic studies because they are uniparentally inherited and relatively slow to evolve. And so they maintain a signature of historic events and demographic fluctuations more clearly than nuclear markers, which are biparentally inherited and generally evolve relatively rapidly compared to chloroplast DNA markers. So I will talk a little bit more about the actual markers that I used in the next slide, but starting then with my sampling. So I conducted field sampling for this study between 2010 and 2011, and it's funny because it seems like it was just not that long ago, and I guess that means I'm getting older because it was actually already over 10 years ago. So this was done, I was on the road for about a 5-month stint during one summer of my PhD work, and I was really happy to have the opportunity to travel really most of the eastern United States or northeastern United States for doing my collections. So as I said, I am from Canada, way up here in the boreal forest, and it was really quite interesting and nice for me to be able to do those collections and see really true temperate forest. I quite enjoyed it. So I collected from a total of 31 natural populations. And in general, I aimed to collect from sites where more than one of my study species was present. So where I did opportunistically sample from some sites where there was just a single species present, but generally I was focused on sites where I could collect, yellow birch and paper birch, or yellow birch and sweet birch. And my goal was usually or generally to sample 30 individuals per species per site. So in purple here are the collections that I made, where I have multiple individuals per per population. And any of these yellow dots are collections that I later obtained from the Canadian National Tree Seed Center. So they sent me seed and I germinated it and ground up the poor tiny little emerging seedlings to extract their DNA like a monster. But those I included just to really increase the geographic coverage, because phylogeographic studies really rely on having a wide geographic representation, and where possible you should aim to sample the entire natural range of your study species. So you'll see I have really pretty complete sampling of the range of yellow birch, shown here in this kind of medium gray, and full sampling of the range of Betula lenta, but then my sampling of Betula papyrifera was much more limited. And that's was really just due to budgetary constraints. We didn't have the budget or the time to send me also all the way across Canada, especially, I don't know how many of you have spent a whole lot of time in Canada, but a lot of, I mean, there are forestry road networks, but a lot of these areas can be fairly remote. So it would have been just time and cost prohibitive to do that sampling. And unfortunately, we really don't have good representation of the prairie provinces in our national seed collections. So I wasn't able to get seed collections from those regions. So for laboratory analysis, as I mentioned, I used chloroplast DNA markers. I looked at 6 different regions of non-coding chloroplast DNA, and those were, they're referred to as simple sequence repeats, the regions that I looked at. So 6 different loci, simple sequence repeat loci I looked at. And then I also sequenced a portion of the psbA trnH intergenic spacer. Doesn't matter if the details of that, but basically a number of different loci that I looked at in the chloroplast genome. So what I actually found. So after doing that analysis, what we do is we look at the unique genotype of each of the individuals that we sampled. And because the chloroplast genome is haploid, i.e. it's uniparentally inherited, we refer to genotypes of haploid genomes as haplotypes. So when I'm referring to haplotypes throughout the rest of this presentation, I'm just referring to chloroplast genotypes. So one of the first things that we did was really mapping the distribution of the different chloroplast genotypes to just get a qualitative assessment of some of the different genetic groupings that occurred, because this helps us to understand what the population genetic structure looks like and what the potential lineage groupings may be. So what we found for Betula papyrifera and Betula alleghaniensis, they actually had fairly similar haplotype distributions. Although these maps don't necessarily look the same at first blush, if you only look at the sympatric portion of the distribution of papyrifera here and alleghaniensis here, what you see is that a number of similar patterns emerge. So for papyrifera and alleghaniensis, what we found is that most of the eastern portion of the distribution was dominated by really just a few closely related haplotypes, which are shown here in purple and orange. Okay. And then we found something slightly different along the Atlantic coastal region here, Nova Scotia, PEI, and a little bit New Brunswick, where a number of populations in that area had a somewhat unrelated haplotype group, which is represented here by the grays and dark greens. And then in those 2 species, we see, again, a similar pattern in the Western Great Lakes region, where those populations were made up of a mixture of 2 different fairly closely related haplotypes, which are shown by the bright yellow and bright blue here in the Western Great Lakes, but also a smaller frequency of individuals that exhibit that eastern haplotype in the purple. So again, the patterns were quite similar between those 2 species. The one difference that I would point out, and this is really just due to the differences in the ranges, is that we did find a different haplotype in the southern Appalachian region for Betula alleghaniensis. And it would be unclear as to whether or not that haplotype might have occurred in papyrifera historically, because its range no longer overlaps with this region. So there was this separate haplotype in the southern Appalachian region. So then on to interpreting that in light of the glacial history, really, of these two species. So we did then what's referred to as a spatial clustering analysis, which is really just designed to simplify the patterns that we just looked at to group genotypes into closely related groups based on how basically how distantly related they are, but also based on their geographic proximity. So that spatial clustering analysis for paper birch revealed 5 different clusters within the range of that species. So there was one group here, actually kind of 2 groups in the Western Great Lakes, one in the sort of extreme Western Great Lakes area, and then another that kind of overlaps with the north shore of Lake Superior and into the Upper Peninsula and Wisconsin. And then this third group, which occurs really throughout the remainder of the eastern distribution. The 4th and 5th group occurred in BC, and these are probably groups that colonized from Western refugia, as opposed to these eastern groups, which we expect colonized from eastern refugia. So just before I go more into that, we'll look at the patterns for yellow birch. So yellow birch, the pattern was very similar. 2 main haplotype groups in the Western Great Lakes region. Most of the eastern distribution composed of just a single group. And then the one thing that was a bit different for alleghaniensis was that we found a distinct group along the Atlantic coast. And at the time, that was kind of interesting. So in terms of what this means from a glacial history perspective, we really have to interpret this in light of what's known with the fossil pollen record for birches. So if you'll remember back to that figure that I presented, the fossil pollen maps indicate that birches likely persisted in a central Appalachian refugia at the time of the last glacial maximum. But there is some evidence based on based on macrofossil records. So macrofossils can include things like leaves and fruits, that birches may also have persisted in a Mississippian refugium. So the way that we interpreted this pattern that we observed was that most of the eastern distribution of this species, because it's composed really of one related group of haplotypes, should have been colonized or should represent just one single glacial refugial lineage, which occurred in the refugium location for that lineage would have been in the central Appalachians. And then after the retreat of the glaciers, those populations, or that population, would have expanded northwards and then westwards across the northern Great Lakes. This population here in the Western Great Lakes was a bit more difficult to really nail down the potential source of it. So because we see kind of 2 somewhat unrelated haplotype groups, this could suggest that these occurred from the Mississippian refugium. And then some studies have also suggested that there could have been a low density Midwestern refugium. So potentially these could be 2 lineages representing a Mississippian and a Midwestern refugium. But the other pattern that we could not ignore, this sort of alternate explanation, could be that these westernmost haplotypes actually expanded eastwards from Western refugium across the prairies. And we really couldn't discriminate between those alternative hypotheses without additional population sampling. So that is something that's been on my agenda for quite some time now, to be able to go and get those samples and to really be able to tease that out. But it's hasn't happened yet. And then finally here along the Atlantic coast, this was kind of interesting at the time, because we were like, well, that area was covered in ice at the time of the last glacial maximum. So theoretically, that population or that cluster shouldn't represent a separate lineage, because where would the refugial location have been? Around, or I guess sort of just before the time I started my PhD, I guess just as I was finishing up my master's, a study on jack pine came out from a group in Quebec, Jean Bousquet and colleagues, and they showed a similar pattern in jack pine. And they actually hypothesized that this lineage should actually represent one that originated from an Atlantic coastal refugium, and that probably this refugia was located on an exposed portion of the Atlantic coastal shelf at the time of the last glacial maximum. So many of you will know that sea levels were lower at the time of the last glacial maximum. So areas which are currently underwater may have been exposed. And actually, if you do look at studies which sort of model or infer where sea ice heights at the last glacial maximum, it supports that there was this area of higher elevation which is currently underwater but would have been exposed during the time of the last glacial maximum. So that told us that we weren't crazy, that this pattern really does probably represent a plausible biological scenario where this lineage did originate from that exposed portion of the Atlantic coastal shelf. So that was at the time quite interesting. Now it's kind of old news. There's been a number of additional studies which sort of support that multiple temperate tree species may have occurred in that Atlantic coastal shelf region. So for Betula lenta, we found a very different pattern. Betula lenta, we found just one haplotype throughout the entire range. And that was really surprising at first, because temperate tree species tend to have very high levels of genetic diversity. So there's that classic allozyme study of Hamrick and colleagues where they related levels of genetic diversity in woody plant species to a variety of different sort of traits. And they generally found that a wide geographic range and a high dispersal ability, i.e. wind dispersed seeds and pollen, are generally correlated to high levels of genetic diversity. So Betula lenta, being a species that has a wide geographic range and has wind dispersed seeds and pollen, our a priori hypothesis was that Betula lenta should have had high levels of haplotype diversity, and probably generally similar to what we would have seen in alleghaniensis or alleghaniensis or papyrifera, maybe a little bit less, but shouldn't have been such low genetic diversity. And in this case, low genetic diversity is a misnomer, because this is actually no genetic diversity. All individuals were genetically identical, at least at the 7 loci that we examined. On further sort of analysis, this pattern actually did make a fair bit of sense. So if you look at the geographic range of Betula lenta and the likely refugial locations of birches during the last glacial maximum, you'll recall that birches are assumed to have persisted within an Appalachian refugia. So if you look at where migration might have occurred from, you would have had migration kind of northwards and southwards out of that refugia. But if Betula lenta was confined to just one single glacial refugia, as we hypothesize, what that means is, from a genetic perspective, that species would have much lower genetic diversity than the other 2 species, because it underwent a more significant population genetic bottleneck. And generally what happens is that you'll find individual refugia becoming fixed for different haplotypes. And that's why in yellow birch and paper birch, we observed 3 or 4 different main haplotype groups, and that's associated with this likelihood that they persisted in 3 or 4 different refugia at the time of the last glacial maximum. So each refugia became fixed for its own haplotype or closely related group of haplotypes. So this does suggest that Betula lenta persisted within just one glacial refugia at the time of the last glacial maximum. And that is why we see just a single haplotype in this species. So in terms of then overall kind of takeaway messages, some of the interesting things that we found, as I said, was the relatively low genetic diversity of Betula lenta was unexpected. The high genetic diversity of Betula, papyrifera, and alleghaniensis was expected because they are relatively broadly distributed species, and they have high cold tolerance, and also because of their polyploid origin. So maybe just before I talk about the allopolyploid origins, I'll talk about those other two sort of traits which we expect should confer are generally associated with high genetic diversity. So I mentioned the study of Hamrick and colleagues, which showed fairly clearly in woody plant species a significant positive correlation between range size and genetic diversity. And so the reason why range size is expected to correlate positively to genetic diversity is that species with larger ranges should have larger population sizes. And large populations lose genetic diversity more slowly due to drift than do relatively small populations. So we expect that the association between distribution and genetic diversity is due to the probability of a higher effective population size in those more broadly distributed species than relatively more narrowly distributed species, which should have smaller effective population sizes. There have been a number of phylogeographic studies of temperate tree species, which have shown a positive correlation between cold tolerance and genetic diversity. So for example, Huertz et al. 2006, if I recall correctly, it's been a while now, they studied European ash species, and they showed that I think the two most cold tolerant species of the three, I believe, that they looked at had a higher genetic diversity, and they hypothesized that that was because those species occurred across a broader geographic range during the last glacial maximum, and so should have suffered less genetic drift than the relatively less cold tolerant species. And then finally, papyrifera and alleghaniensis are both polyploid species. As I mentioned, they're hexaploids, and they are both believed to be allopolyploid in origin. So for those of you who may not be familiar with sort of like types of polyploids, so a polyploid is basically just any species that has more than two alleles at any nuclear DNA locus. So in this case, they both have 6 alleles. So polyploids can form in a number of ways. They can form either by genomes, what's referred to as spontaneous genome doubling in one species. So for example, there's an ancestral species and something, there's a failure of meiosis, and you get an unreduced gamete forming with, uniting with another unreduced gamete, and that could result in polyploidy resulting not from hybridization, but just genome doubling within a species. What's believed to be more common in plants is polyploidy that results from hybridization. So allopolyploidy, allo basically meaning other, is polyploidy that is thought to result by hybridization between separate species. So both alleghaniensis and papyrifera, there's genomic evidence to suggest that they originated from hybridization between two ancestral species, and so they maintain the genetic diversity of each of those ancestral species within their contemporary genomes. So by mixing up essentially more ancestral tree species, you get a greater genetic diversity. And there are enough, like theoretically, that's been shown and predicted quite well that allopolyploids should have relatively high genetic diversity compared to autopolyploids or diploids. But there are a number of empirical studies that seem to point to that as well, such as that of Nienberg et al. So the haplotype sharing was something that was really unexpected between papyrifera and alleghaniensis. So we observed that most of the haplotypes that we observed in these species were actually shared between species. So when we looked at the sites where papyrifera and alleghaniensis co-occurred, there was a total of, I think, 17 sites where these two species co-occurred. And what we found was that there was a total of 8 haplotypes across in both species at those 17 sites, and that 5 of them were shared. Only 3 of those haplotypes were unique to one species or the other. So we did calculate the statistic, which is known as an introgression ratio, and it's basically just a measure of how similar the haplotype frequencies are among species at the same sites. And the value ranges between 0 and 1 meaning that they're basically the haplotype frequencies are identical at shared sites. And we found an introgression ratio of 0.98. So essentially, the haplotype frequencies at shared sites were basically identical, close to identical. Which means that haplotype sharing isn't random, it has a strong geographic pattern. So there's a number of different sort of evolutionary mechanisms that can result in haplotype sharing. And just as sort of full disclosure, before we started this study, we did not expect to see this kind of a pattern. We expected Betula papyrifera to kind of have its own haplotypes and Betula alleghaniensis to kind of have its haplotypes. We did not expect to see this level of haplotype sharing. So that led us to the question, and also reviewers, when we did finally submit this paper, to really question what the likely evolutionary origin of this haplotype sharing was. And there's three possible sources of haplotype sharing among species. And those are homoplasy, that's where you get the same mutation arising in different species by chance. There's also shared ancestry, where individuals have the same alleles because they are closely related, so closely related by descent. They share a recent common ancestor. And that's also referred to as incomplete lineage sorting. And then finally, the third sort of explanation is introgression. And the possible explanations of homoplasy and shared ancestry were rejected. We rejected them outright based on a number of statistical tests that we conducted. But basically, if you look at homoplasy and shared ancestry, they are expected to result in a random pattern of allele sharing, where you would expect maybe both species would have this yellow haplotype, but it might occur here in papyrifera and it might occur in these populations in alleghaniensis. But that's not what we found. We found this like geographically structured pattern of haplotype sharing, which is more concordant, or really only reasonably explained by introgression, by historic hybridization between these two species. So that was the and that is the sort of explanation that we have taken from this. So we do consider that this allele sharing results from historic hybridization between these two species. And our inference is that because of the observed pattern and just the properties of chloroplast DNA markers, that this observed pattern of haplotype sharing should represent hybridization that occurred historically between these two species. So that hybridization would have occurred between these two species within glacial refugia. So for example, let's assume that both papyrifera and alleghaniensis inhabited this Mississippian refugia. That they would have hybridized within that refugia, resulting in haplotype basically capture, transfer between species. And then after they migrated northward, you see the similar pattern that both of those haplotypes that would have been present in that refugia would be reflected then in the contemporary populations. So we consider this to represent historical hybridization between these two species. And there are quite a number of studies, a growing number, which have found similar patterns, the European oaks being kind of the example of very widespread haplotype sharing, where basically the genetic makeup of a species is better predicted by its geography than by its species, because there's such a high level of historic hybridization that has occurred between many of the European oak species. And that work was done by really Petit et al. In kind of the late 90s, early 2000s, and was quite groundbreaking at the time. So the second part of my study, and I'll try and go fairly quick because I know we're getting to time here, and this is much shorter, I assure you. But what we wanted to do for the second part of this study was that once we recognized that there was this high level of allele sharing, we wanted to better understand whether there might also be contemporary introgression or contemporary hybridization between alleghaniensis, and papyrifera. So the second part of my study looks really at introgression at nuclear genes. So there are a number of studies which have shown that introgression, this should say at chloroplast markers, is not necessarily associated with introgression at nuclear DNA markers. So remember, plants have those two different, well, actually three different genomes, but chloroplast versus the nuclear genome have different properties. And because of that, you can find or detect chloroplast introgression in some cases where there is not really much or any evidence of contemporary nuclear DNA introgression. And I'll talk a little bit more about how that can happen, sort of the mechanism that explains that later on. As I mentioned, the chloroplast DNA marker is uniparentally inherited. In angiosperms, it's generally maternally inherited, so inherited clonally from the mother, whereas nuclear markers are biparentally inherited. And nuclear markers are generally better at detecting contemporary hybridization than chloroplast DNA markers. Chloroplast DNA markers can carry signals of historic introgression for a really long time, whereas nuclear markers, because of that biparental inheritance, the signature of historic selection kind of gets mixed up and gets sort of blurred out by recombination. So there was a number of reasons why we expected that we could find evidence of natural hybridization in birches, as seen by nuclear and microsatellite allele sharing. And that's because natural hybridization is really frequent among Betula species. So there was a phylogenetic study of De Young in 1993, and he actually did a sort of a circumscription of the genus, and he showed really significant natural hybridization. And there's been quite a number of controlled cross studies done, and those have shown even that North American birches will often hybridize very readily with European birches, which obviously would have diverged quite a while ago in terms of their evolutionary history. And that also you can find hybridization between basically different lineages within the family. So the dark bark birches are generally form kind of one clade in the phylogeny. So dark bark birches includes obviously yellow birch and sweet birch, those with darker bark. And then there's the light barked birches, which generally includes like papyrifera, populifolia, nana, which is a European species, pubescens, which is a European species. But those are quite distantly related. If you look at species in the dark bark clade versus the light bark clade, they're expected to have diverged millions of years ago, but controlled cross studies still show that they can be cross compatible, which is quite remarkable because normally natural hybridization only occurs between species that share quite a recent common ancestor, so usually sister species. There is evidence from a number of studies that Betula alleghaniensis and papyrifera do hybridize naturally based on the finding of morphological intermediates. So, for example, the studies of Barnes et al. And Clausen found morphological intermediates. And then also there's been a number of controlled cross studies which have shown that Betula alleghaniensis and lenta are cross compatible. You can generate hybrids from matings between those two species. And natural hybrids have been expected to occur between those species for a long time because they're co-distributed, so the range of lenta is completely sympatric with the range of alleghaniensis, and also because they overlap in flowering time. So you would expect that significant natural hybridization should occur between these species. So we had a number of reasons to expect that we should find evidence for hybridization. So for this particular part of the study, we looked at a subset of our populations that we use for the phylogeographic study. So in this case, I only I sort of thinned out any sites that had less than 20 individuals per species. And that's because for nuclear markers, you need larger sample sizes to make accurate inferences. So in total, we had then 20 sites with a minimum sample size of 20 individuals per species. And this map is a bit of a mess, but basically it just shows the distribution of each of the species in different colored outlines, and then the sampling locations. So we had locations throughout the Western Great Lakes region, not really anything through Ontario and Southern Quebec, because we only had like small population samples in those regions, but then good sampling throughout the Appalachian region and the northeastern United States. So for this particular study, as I mentioned, I used nuclear DNA markers, and specifically portions of the non-coding nuclear DNA, which have short tandem repeats, which are referred to as microsatellites. So I looked at 8 different loci, neutral DNA loci. And this is just an image of basically the peaks that we use to analyze the genotypes of each individual. I won't go into all of it, but sometimes I show that from some of my students because they find it interesting, but I'd be happy to talk about it in questions, but for the sake of time, I'll skip over it. So what we found when we analyzed the frequency of those nuclear DNA markers for each species, so we looked at the frequency of each marker in each population of each species, and then did a principal components analysis on that to basically look at how genetic variation was partitioned among the species, the sort of putative species groups. So what we found was that there was relatively clear separation of the species. So Betula alleghaniensis individuals are shown here in red, Betula papyrifera individuals are shown in blue, and then Betula lenta individuals are shown in green. And what we saw was that they formed relatively distinct genetic clusters. This suggests that they are largely differentiated in terms of their allele frequencies. We did find a number of individuals that were kind of in the middle of that psbA space, which could represent potential hybrids. So there were a number of intermediates in terms of their genotype makeup, their genetic makeup, that could represent hybrids between those species. So then we did another analysis, which really just looks at, again, the overall genotype frequencies within populations, but specifically the genotypes of individuals. And it's a Bayesian analysis, which basically groups individuals into populations to kind of bring them into Hardy-Weinberg equilibrium. So it's equilibrium. It's a method of delineating natural populations, or basically contiguous populations from a genetic standpoint, as opposed to when I go out and sample 10 sites, that might actually represent 3 populations if those sites which I sampled are connected by gene flow. Okay, so there's a difference between sampling sites versus like populations from a genetic standpoint. So this structure analysis that we did was just intended to identify the genetic clustering of individuals within these 3 species groups. And what we found was that I'll explain this briefly. So for each species, this is basically a bar graph, even though you can't see it. So there's basically hundreds of individual bars here, and each bar represents an individual. So if one individual here is all yellow, what that means is basically all of the alleles in that individual are inferred to have come from its representative species cluster. So for Betula alleghaniensis, it's represented by yellow. So seeing in a number of individuals some blue and pink bars means that there's a number of alleghaniensis individuals that were inferred to carry alleles which were more typical of papyrifera and lenta. So these could be potentially admixed or hybrid individuals. You then look at that separately for each species. So for papyrifera, we found generally that individuals that were identified more based on their morphology as papyrifera tended to have a more pure genetic makeup. A few individuals of mixed ancestry were identified, but obviously a much lower proportion. And for Betula lenta, again, most individuals had a high, most of their alleles inferred to have come from the lenta species cluster, but there was a few individuals that had alleghaniensis type or alleghaniensis-like genetic makeup. So most individuals were not admixed, but we did identify a number of alleghaniensis individuals as having admixed ancestry. So basically potentially hybrids between alleghaniensis and papyrifera and between alleghaniensis and lenta. So we basically mapped that genetic clustering that we did in structure that I showed you on the previous slide to look at geographic patterns of allele sharing. So here for Betula alleghaniensis, each of these pies represents basically the individual genetic makeup of individuals in that population. So for alleghaniensis, it's represented by yellow. So the larger the pie, or the larger the portion of this circle that comes from yellow, means that more individuals in that population were inferred to have basically like pure alleghaniensis type alleles. And then blue indicates some alleles in individuals in that population which were inferred to have originated from lenta. And then pink indicates that some individuals in this population carried alleles that were inferred to have originated from papyrifera. So what we can see is that basically throughout most of the range of alleghaniensis, the kind of admixture proportions are more or less constant, with most alleles inferred to belong to alleghaniensis, and then a small proportion of individuals harboring some lenta type alleles. In the Western Great Lakes region, though, the pattern's a little bit different. Here we see some Betula papyrifera alleles creeping in, but again, we still see this pattern of a number of individuals having lenta type alleles. So we did observe a significant pattern of admixture with respect to latitude. So we did a number of additional admixture analysis, and basically what we found was there was a significant correlation between how pure the makeup of a species was compared to the latitude. And we found that obviously, as shown here, that populations from the Western Great Lakes region at higher latitudes had higher admixture proportions. So in other words, more individuals in these populations were inferred to be of hybrid origin than individuals in southern populations. We did not find a significant trend in papyrifera and lenta. In papyrifera, we found that really basically there was a small number of alleles which were inferred to have originated from alleghaniensis, but they were more or less constant across the range, and the same for lenta. So we inferred then that this pattern resulted from contemporary hybridization between alleghaniensis and papyrifera. So this should represent actually basically capture of papyrifera type alleles into alleghaniensis through hybridization. I think we're okay. Yes, we're on the last few slides, so I'm wrapping it up. Sorry, guys, it's a bit long, but I pruned it down, but it's still long. So the high proportion of shared alleles, we inferred that some allele sharing between papyrifera and alleghaniensis may have originated from shared ancestry, but it's not super likely because those two species are quite distantly phylogenetically related. But the geographic pattern was really consistent with the fact that allele sharing that we observed between papyrifera and alleghaniensis should originate from contemporary hybridization as opposed to shared ancestry or homoplasy, as I mentioned in the previous section. And this is not, I guess, unexpected. There are a number of studies which have found morphological intermediates of alleghaniensis and papyrifera in the Western Great Lakes region. That's also where I found a number of morphological intermediates in my fieldwork, and I didn't find any morphological intermediates for these two species in the more eastern and southern distribution of papyrifera. So it kind of agrees with what the patterns that have been observed based on morphology. There's been studies which have shown that in the Western Great Lakes region, the flowering times of these two species overlap sufficiently such that cross-pollination and hybridization could be expected to occur. So the Western Great Lakes region appears to represent a zone of natural hybridization between yellow and paper birch. On the other hand, alleghaniensis and lenta, they did share alleles, but there was no geographic pattern to the allele sharing. The number or the proportion of alleles in alleghaniensis that were inferred to have originated from lenta was more or less constant across its distribution. So that is indicative that allele sharing should arise either from homoplasy or shared ancestral polymorphism. So our hypothesis is that this allele sharing between alleghaniensis and lenta has occurred due to their shared ancestry. They're closely phylogenetically related. A number of studies have inferred them to be sister species. So it's probable that they might carry those shared alleles from a shared recent common ancestor, as opposed to sharing them because of hybridization. And so this geographic pattern is more consistent with allele sharing due to shared ancestry. So then in conclusion, the three species are genetically distinct at nuclear DNA markers, despite high levels of chloroplast DNA sharing that we observed in the first part of our study. Alleghaniensis and lenta appear to share alleles due to shared ancestral polymorphisms, whereas alleghaniensis and papyrifera, the geographic pattern of allele sharing is suggestive of introgression, and in this case, at nuclear markers, we're inferring that historic introgression has occurred. So based on these two studies, we infer that both historic and contemporary introgression have contributed to allele sharing between papyrifera and alleghaniensis. And we infer that the introgression, historic introgression likely occurred within glacial refugia or during post-glacial migration, where pollen of Betula papyrifera would have, so after the retreat of the glaciers, Betula papyrifera was the first to colonize northwards based on macrofossil evidence. So we can infer that papyrifera was the first to colonize northwards. So it's expected that papyrifera pollen may have then landed on those northward expanding, or sorry, alleghaniensis pollen would have landed on those northward expanding papyrifera pollen populations, resulting in the formation of hybrid individuals. If those hybrid individuals then back-crossed with yellow birch, i.e. mated with the parental yellow birch population, what would happen is that you would get a nuclear genome of Betula papyrifera with a chloroplast genome of papyrifera with a nuclear genome that is more consistent with yellow birch. So that's why we can find these kind of differing patterns between chloroplast markers and nuclear markers, is through this uniparental versus biparental inheritance and associated backcrossing with parental species. So I hope that wasn't too complicated, but I think we have a bit of time for questions.
Awesome. Thank you so much, Ashley. I know that for me, this was really interesting because the what I'm doing, the research I'm doing for my PhD is on larix, larch, which is a deciduous conifer, but that has some of these similar kind of things going on, which I think most tree species, probably almost all tree species in the boreal forest, have this much more recent kind of changes in their the populations and where they are. So it's really fascinating, in comparison to more tropical or lower temperate regions where, you think about, oh, they have these kind of really long standing kind of patterns of where the species are located for the phylogeography, whereas all of the things that were glaciated, it's really the last 10,000 years that are really being exhibited right now, which is really cool.
Absolutely, yeah.
So definitely have, give me some more things to think about with my own research too. I hope that I can get, I would be really happy to see some kind of these kind of like pretty interesting and clear kind of patterns, that and another thing that in the conifers that gives it another level of kind of interesting things is that the chloroplast genome is paternally inherited. So then we have the mitochondria are maternally inherited, the chloroplast is paternal, and then the nuclear is. So there's even more kind of potential for kind of conflicting stories in these three different things, and how pollen can be dispersed, how far the pollen will be dispersed versus how far the seeds will be dispersed, kind of. So
Yeah, there was a really interesting study. I mean, again, it's old now, but by Jean Bousquet and colleagues on the Picea chihuahuana, where they use the chloroplast and mitochondrial markers for disentangling seed versus pollen flow. And I thought, oh, that's really interesting. Yeah, it's really nice to be able to do that in the conifers for sure.
Yeah, and especially now, it's also interesting to see, I guess, how much the kind of DNA technology and the amount of data that we're getting now has been like kind of exponentially increasing. That people are looking at doing entire chloroplast sequencing. Of course, funding is always still an issue, but that there's so much that the inference can be even more kind of backed up, that sometimes it will probably be the same patterns will be uncovered whether you're using 5 loci versus the entire genome. But just even just for statistics, it's really
It is, and that's one thing I've always wondered. If we were to go back and look at some lenta with like rad seed data, would we find the same pattern? And so, like, that's also on the agenda for the future. Would we find one haplotype, or is it just that we didn't have the resolution with those chloroplast markers? So,
right.
Yeah.
All right. If anybody has any questions from the audience, please feel free any way that you would like. But it's also, I guess, interesting to hear about. I know, so alleghaniensis around here is relatively rare. I think it's only, at least officially, recorded only in the Bronx, and there's a few that are in the old growth forest here that we have in the botanical garden. That's the only place I've seen it. And so, I guess especially given it's reflected in its name, that it's much more kind of found in the Appalachian Mountains.
Yeah, and that's definitely where it was like very high density for sure in my sampling. And I know like even like northern kind of Michigan, Wisconsin, like I really had to struggle to be able to find the pockets of it for sampling. But yeah, definitely Appalachians is like it's everywhere. You don't even have to try. It's absolutely everywhere.
And I guess also when you were doing that, the field work, what was the process like? Was it more you knew, okay, I know I'm going to go to this specific place based on there's been some herbarium collections there, or someone told me, or was it just kind of let's go on a search and search for birch?
Yeah, it was search for birch. Yeah, it was I guess sort of random. So basically my main I guess my kind of first step was to kind of look at the natural distribution and identify national and state parks where I would have a likelihood of collecting and not have somebody threatening to shoot me for wandering onto their property. Of course, as a Canadian, you always have this idea that if you're going onto somebody's property in the US that you might get shot. So that was in my mind, but like definitely, yeah, identifying the state and national forests where there would be a likelihood of being able to collect the number of individuals, because often you need to kind of traverse a few kilometers of road, because I did have to make sure my samples were between 50 and 200 meters apart, because otherwise you're likely sampling the same maternal tree. So depending on this kind of the conditions in that area, you may have to kind of travel the road network in that area a little bit. So yeah, if you're just looking at one little piece of property, you wouldn't be able to get the amount of samples you needed. Yeah, and then it really just came down to like where I could get the permits in time to do the sampling. That's again something that I was just not like familiar with, because we have so much crown land in Canada. It's like you just go out and start cutting leaves and it's not a problem. But to realize that, oh, I need a permit to be able to do that was a surprise for me.
And then everything's kind of parceled and owned by a different entity, even if it's all,
that was
public. It's like what's from the state, what's federal, and what's
absolutely. So it was all a big learning curve for sure. So
yeah, but that's also really awesome to have been able to kind of traverse all of that space and kind of
it was
see.
amazing. So I had a tiny little vehicle, which was, I don't know if you know the Toyota Yaris, but it's just a tiny little vehicle. That was my car, and it was leased, and that was my field vehicle. So I had to fit my like pruning poles and everything. I was camping. I wasn't staying in hotels, so I had my tent and all my camping gear and everything in that tiny little car, and that was my off-road vehicle for driving the bush roads of the Appalachians. And I was like, oh, I'm gonna die. I'm gonna get stuck and I'm gonna die because no one knows where I am. So
When
yeah,
they
it was
returned the lease,
interesting.
I'm sure they were real happy with.
Yeah. Yeah, it was a cool experience and beautiful, beautiful countryside. So
Awesome. Well, thanks so much for sharing this this research with us. And again, anybody who came in a little bit late, if you're interested, we'll have it posted on our YouTube page soon, so you can check it out there. And if you want to get more down into the nitty gritty, there are publications. There's two publications, if I'm not mistaken,
yeah.
on these that go more into depth. If you're interested in looking at that, or seeing some of those figures a little bit bigger on maps and all things like that.
Yeah, and you can always email me too. I'm always happy to talk about birches. They're just passion. So yeah.
Yes, awesome. Well,
Awesome.
thanks so much again. Thanks everybody for coming. And stay tuned. Next month, we will have a talk probably about plant herbivory defense. So keep an eye out for that. So stay well, everybody. We're supposed to have a bit of a winter storm overnight here. So everybody stay warm, and we'll see you soon.
Thanks.
Bye-bye.